Hello everyone. I welcome you all to the fourth lecture of our course, Cellular Microenvironment from Oxygen Gradient to Gene Expression. In previous lecture, we already learned how a cell sense low oxygen through the hypoxia-inducible factor or HIF. We also discussed that under normal oxygen condition, HIF-1 alpha is continuously degraded and that therefore cannot activate gene expression. However, when oxygen level falls, HIF-1 alpha becomes stabilized and that enters into the nucleus and that combines with the HIF-1 beta, that is another protein, and that is consistently present inside the nucleus and that binds to a specific sequence of the DNA, which is known as the hypoxia responsive element. And the short form of this is HREs. So this ultimately activates the transcription of several genes that helps the cell adaptation and survival of the cells under hypoxic condition. So at this point, it may seem that we have completely understood oxygen sensing, but there is a very interesting question that still remains unanswered. Uh, if HIF-1 alpha is capable of regulating the cellular response to hypoxia, then why do cells also contain another oxygen sensitive transcription factor, which is called as the HIF-2 alpha? And why does nature need two different master regulator for oxygen sensing? Do they perform the same function or do they regulate different biological processes? So in today's lecture, we will answer these questions by comparing HIF-1 alpha and HIF-2 alpha. We will also understand that their structural similarities, their important differences, the gene they regulate, and why both proteins are essential for maintaining the cellular adaptation under different hypoxic conditions. So let's begin our discussion on the two master regulator of oxygen sensing, that is HIF-1 alpha and HIF-2 alpha that you can see here. This is the fourth lecture of this course. And this is very interesting. So we will slowly we will be going in the deeper concept of understanding the gene expression analysis, right? So if we talk about why HIF-1 alpha is important for the cancer, so when we talk about the solid tumor, that grows faster than their blood supply, and if you see here that abnormal and insufficient blood vasculatures that cannot deliver the enough oxygen to the cells. Right. So based on this, if you see that if this is a blood capillary through which the oxygen is getting transported through the red blood cells, and due to the limited diffusion of oxygen, nutrients, and other metabolites, you can see that these solid tumor has been divided into three different regions. And already I have discussed these things in the previous lecture also. There is a proliferating zone, quasicate zone, and necrotic and hypoxic core. So if you have not understood this concept very well, then just go to the lecture number 3. Then you will be able to understand these things in best possible way. So as we have seen that you can see in solid tumor there is a irregular or leaky blood vasculature, and that cannot support the transport of oxygen. And in limited condition of oxygen and in presence of oxygen, we have seen that how HIF-1 alpha is getting stabilized and destabilized. So when we talk about normal oxygen condition, that is called as the normoxia. So in normal oxidant condition, we have seen that the PSG enzyme basically, hydroxylate HIF-1 alpha, and basically it adds the alcoholic functional group over HIF-1 alpha. So I'm not going to explain about the chemistry part of this particular thing, but in another lecture I'll try to explain the whole chemistry, how this hydroxylation reaction is happening over here. And then this hydroxylated HIF-1 alpha is recognized by VHL protein, and now VHL protein basically UV quantize this hydroxylated HIF-1 alpha, and that UV quantized HIF-1 alpha will be recognized by the proteasome. And this is happening in presence of normal oxygen. No, I mean normal oxygen condition, normoxia, where only PSG enzyme will work. And then the proteasome basically degrade your HIF-1 alpha. So HIF-1 alpha is hydroxylated in normal condition, and that will be recognized by VHL, and ultimately it will get degraded. But when we talk about the limited oxygen condition where there is hypoxia, in that condition, the PhD enzyme that will not get activated, and due to this inactivation of PhD enzyme, it cannot, hydroxylate your HIF-1 alpha, and unhydroxylate HIF-1 alpha cannot be recognized by VHL protein, and that cannot be ubiquitinize this VHL, hydroxylated HIF-1 alpha, and then proteasome cannot detect these things. So that's why these HIF-1 alpha molecules will start getting accumulated inside the cells. So this is how the stabilization and degradation of HIF-1 alpha takes place inside the cells. Now coming to the very important thing that uh, due to the let's say in presence of limited oxygen in hypoxia, HIF-1 alpha is stabilized. Now this HIF-1 alpha will start accumulated inside the cell, and finally it will start getting translocated inside the nucleus. And once it gets translocated inside the nucleus, it binds with the HIF-1 beta protein. I'll discuss about HIF-1 alpha, why there is HIF-1 alpha, why there is HIF-1 beta, not HIF-2 beta, why there is HIF-1 alpha, HIF-2 alpha, not another thing, something like that. Okay, all these things I'll discuss in the next lecture, but here just try to understand now. This HIF-1 alpha and HIF-1 beta will form the dimer, and that is called as the transcription complex. And now this transcription complex ultimately enters inside the nucleus, and then it binds to the specific sequence of the DNA, and that is called as the HRE, hypoxia responsive element. And that basically promotes your angiogenesis, that is VEGF expression, that will see so the VEGF overexpression there. So this promotes angiogenesis means that basically give a signal for the new blood vasculature formations to increase the oxygen and nutrient supply. Then it also alters cells metabolism, that increases the GLUT expression, glycolytic enzyme, that increases the GLUT uptake and shift metabolism towards glycolysis to produce energy even in the low oxygen condition. Then there is a that also supports the cell survival pathway, and it enhances the tumor progression like invasion, metastasis, and therapeutic resistance. So if you see the solid tumor due to this particular region, you will see that different region of the solid tumor are like proliferating zone, quiescent zone, and necrotic code that shows different kind of resistance for any kind of chemotherapeutic reagent. So that's why that promotes the invasion, metastasis, and resistance to the chemotherapy and the radiotherapy. So ultimately, you can see that that supports the tumor growth and aggressiveness in absence of oxygen also. And that is what the HIF-1 alpha driven adaptation enables the cancer cells to thrive or survive, grow, and spreads despite of the limited oxygen condition. Now coming to the next part, that is very important part of this lecture, why cells need to hypoxia inducible factor, that is HIF-1 alpha and HIF-2 alpha, not only HIF-1 alpha or not only HIF-2 alpha explicitly. And that is what we are going to answer in this slide. So if we talk about limited oxygen condition, let's say if a cell sense the limited oxygen condition, then what happens? The HIF-1 alpha molecules cannot get degraded by the proteasome. So now what will happen? This HIF-1 alpha inside the nucleus, once the HIF-1 alpha is getting translocated inside the nucleus, it binds with the HIF-1 beta, and that forms the HIF-1 alpha, HIF-1 beta dimer. Right, and this dimerization takes place inside the nucleus, and now this complex that is called as the transcription complex that binds to the specific region of the DNA that is called as the hypoxia responsive element, and then that basically target uh, gene transcription activated. So this basically activates the target gene. So now coming to the why HIF-1 alpha. So when we talk about HIF-1 alpha, that basically known as the early responder. That means it get overexpressed or it get expressed when there is a limited oxygen for the very small time. Like that is called as the acute hypoxia condition. That means it gets activated within minute to hour. And what are the genes that is getting activated due to the HIF-1 alpha? That is the glycolysis, glucose uptake, anaerobic metabolism, lactate production, and short-term survival, right? So cells will try to survive in that condition due to the limited oxygen condition for the for the very small time, and for a very small time, all of these genes are getting activated. Next thing is that the key target genes will be GLUT1, HK2, LDHA, and PDK1. So I'm not going in the details of all of these things. Obviously, this will take very long, lecture to complete all of these genes. Just I wanted to let you know the key target gene that is getting activated in presence of if cells experiences limited oxygen for a minute to hour condition. Then what kind of tissue in which kind of tissue this hypoxia or HIF-1 alpha getting expressed, like that HIF-1 alpha is getting expressed in nearly all cell type, that is called as the universal hypoxia responder. Now, coming to the another hypoxia inducible factor, that is HIF-2 alpha. So, why HIF-1 alpha and HIF-2 alpha? In next lecture, I'll discuss these things in detail. But for very fundamental understanding, I'm just letting you know. So, scientists first discovered HIF-1 HIF. So that's why they have put the nomenclature as HIF-1 alpha, and subsequently again they discovered another hypoxia inducible factor, and the name is HIF-2 alpha. So that is based on the chronology only. So, when we talk about the HIF-2 alpha, that is known as the late responder that is getting activated or expressed under chronic hypoxia condition if cells, cells are getting exposed for a prolonged hypoxia that will take from hours to delay. And what are the genes that is getting activated in this long oxygen deficient condition? That is angiogenesis, erythropoiesis, cell proliferation, stem cell maintenance, then long-term adaptation, and the tissue remodeling. So these are the genes that will get activated. And what are the key target genes? Again, VEGF, EPO, OCT4, and cyclin D1. I'm not going in the details of all of these genes. Just this is the key factor. And then it's important to understand what kind of, HIF-2 alpha, in which tissue this HIF-2 alpha is getting expressed. So, like you can see that endothelium tissue, kidney, liver, lungs, and heart, certain stem cells also it is getting expressed in, prolonged hypoxia condition. Now, coming to the very important part of this HIF-1 alpha and HIF-2 alpha. So, when scientists say that HIF-1 alpha and HIF-2 alpha are structurally similar, so they do not mean they have exactly the same amino acid sequence. Instead, they mean they, contain almost the same domain. These domain appear nearly in the same order, and each corresponding domain performs, almost the same basis function. So if we see the structure of HIF-1 alpha, so as I've told you, this is a kind of protein, and the protein is made up of, sequence of amino acid, and this amino acid sequence will be having N-terminal and C-terminal, and that you can see in this structure. So all of these amino acid will work differently, like BHLH protein that basically, amino acid that is basically responsible for DNA binding. So as we have seen that when HIF-1 alpha and is getting translocated inside the nucleus, it basically binds with the HIF-1 beta, and then this HIF-1 alpha and HIF-1 beta forms a transcription complex, and that will binds with the a specific sequence of the DNA that is called as the hypoxia responsive element. So that binding with the DNA is facilitated by BHLH amino acid sequence. Then PASA and PASB is responsible for the dimerization with HIF-1 beta, as we have seen earlier. Then the oxygen sensing, as we have seen that in presence of oxygen only, PSD PSD enzyme will start working. And for oxygen sensing, there is a specific sequence of amino acid that is the ODDD. And then the transcriptional activation, that as I have told you, that the gene which is getting expressed inside the DNA that should get transcripted into the mRNA, and that has been performed using, TADN and TADC. And so we can see here there is an N terminal and C terminal of the protein. Now coming to the HIF-2 alpha, basically you see there is no difference between the structure in the structure of HIF-1 alpha and HIF-2 beta. It also contains an N-C terminal. It also contains specifically the same, base amino acid sequence like BHLH, PASA, PASB, ODDD, TAND, and TAND. Similar structurally, they both are similar, but as I have told you, they contains almost the same domain, but these domain appear in nearly the same order. So every time it cannot get expressed, right? So as I've told you, if there is a acute hypoxia, then only all of these HIF-1 alpha will get expressed, and if there is a prolonged hypoxia or chronic hypoxia, then only HIF-2 alpha will get expressed, right? So when we talk about the HIF-1 alpha and HIF-2 alpha, we have already seen that there are different roles at different stages for a hypoxia response. Like HIF-1 alpha is like, that handles the emergency. So keeps the cells alive during the sudden oxygen shortage. And obviously cells are having, good feedback mechanism. So there are all of these things would be there inside the cells so that it can regulate the survival pathway, or it can activate the survival pathway. And if it sustain for a very long period of time, if let's say cells feels oxygen shortage for a very long period of time, then plan B comes, that is HIF-2 alpha, that enables long-term adaptation, growth, and the survival. Okay, so I'll stop this lecture here, and before stopping, I'll summarize this lecture that we understood why cells require two hypoxia inducible factor, that is HIF-1 alpha and HIF-2 alpha. The motivation is clear to all of you that both of the working principle of both of these things are the different. One works in the certain oxygen shortage, and another works for long-term adaptation of cells growth and survival in presence of prolonged oxygen deficiency. Okay, so this is all about this lecture. In next lecture, we will understand another very important part, that is what do you mean by the gene expression, especially because repeatedly I talked about there is a gene expression. So in next lecture, we'll understand what does gene expression mean actually. Okay, so I'll stop this lecture here. See you in the next lecture. Thank you.